So I am starting the recording. Welcome everybody. It's Wednesday. We are in the middle of week 7. We are moving into more of our lymphatic and immunity systems. So we left off on, we've gotten through how big concept, lymphatic vessels and cardiovascular vessels. Make those comparisons. How were they the same? How were they different? Where are the differences and why? Okay. Then we went through what are the organs of the lymphatic system, what are the tissues of the lymphatic system. And again, you have to a little bit of look for the capsule, look for the blood vessel supply, and again for the thymus gland, it's not really going to be part of the immune system function in a sense of it's actively engaged in getting rid of a pathogen inside of it, but it's part of the process of making T cells mature. Okay, and we'll hit a little bit of that more today. So when we talk about the immune and the lymphatic system, we are looking for where are there a large population of lymphocytes. And remember, lymphocytes are T cells, B cells, and natural killer cells. And they tend to exist and clump together in these lymphatic tissues, lymphatic organs. And we will also see present with them, in many cases, eosinophils, mast cells, neutrophils, and macrophages. So a lot of our immune cells are going to end up together with the lymphatic cells. Okay. And the reason why they're all together is they all have a certain role through the immune process and they depend upon each other. So I need some macrophages, I need some neutrophils, I need some eosinophils, I need some mast cells, because those are how I break apart pathogens that I come into contact to, and those are how I, not necessarily for mast cells, but for the others, those are how I phagocytose pieces and parts of cells or full cells and bring them and present them to my T cells. All right, so my T cells want to be in near contact where I might have a lot of macrophages, neutrophils, and eosinophils, so they can again be given the opportunity to inspect by presentation on a membrane protein what are the neutrophils, the eosinophils, and the macrophages eating. Okay, we're also going to learn a little bit in the immune system, and it's not very clear in your book, but there are many cells that are macrophage like. All right, dendritic cells are going to be mentioned, and you're going to be like, what is this dendritic cell? And you're going to look a little deeper, and it is a macrophage like cell. Is there a little bit more to the story? Yes, but it isn't quite fleshed out in the research and in the literature, so we're not going to worry about that. We're just going to call it a macrophage like cell, and for our purposes, it is going to be like a macrophage, an antigen presenting cell. When it eats something, it wants to show it to the T cell. Okay, so that is one of the examples of a cell that's mentioned in the lymph node, and it's kind of a surprise. You have this dendritic cell, and if you go back to chapter 4, it was mentioned, and they don't really explain what it is. Okay, but for our purposes, we are going to think of it as a macrophage-like cell, and it, just like a macrophage, will eat something and then present what it eats to T cells to be inspected for, is it self, is it not self, is it something we need to activate against and form a more specific immune response. Okay, all right, so let's talk more about the cells. All right, so lymphocytes, remember your T cells and your B cells, when we looked at them at the naked eye, we were not able to really differentiate what's a T cell, what's a B cell, what's a natural killer cell by looking at the cells in the blood. But we knew that they are the second most popular or the most abundant cells in our blood in circulation. And part of that comes from, well, there's a lot of them. There's a lot of different types of T cells, a lot of different types of B cells, and then we have natural killer cells. And when you clump them together, yeah, there's about, out of 100, there probably will be about 20 of these guys circulating. They could be circulating because for the T cells, they're heading to the thymus gland, maybe to become mature. They could be leaving the thymus gland, leaving the bone marrow, and heading to their location, like lymph nodes, the spleen, the dermis of your skin, the connective tissue, the submucosal layer of your respiratory tract, the submucosal layer, the malts and palps in your digestive tract. So they could be potentially heading to a location, Or they might be just hanging out in the blood, maybe because they're going to be in the blood a few rounds, a few cycles in case something is in the blood and is attacking us. Okay, so there's lots of reasons why they could end up in the blood at any given point in time. And when they are going to be like ants, they are going to be looking for certain signs, certain cytokines, certain molecules that are, again, we don't have it all figured out. But when those signals come, they are going to be able to exit out of the blood supply along with all of our neutrophils. And like the neutrophils, they are going to target an area that we potentially have a cut, we have a scratch, we have a break in the barriers, and they are going to head to that area to help with the process of cleanup, with destruction of debris, with destruction of any pathogen that has tried to take advantage of the cut, of the tear, of the break in skin or the break in the lumen, and is trying to gain access to the inner workings and protective areas that they would then be able to propagate and expand and use the human body as a host. Okay. So that is part of the reason why we expect to see a lot of lymphocytes in our circulation. Now, there are many types of B cells. There are many types of T cells. As of right now, there's one form of natural killer cell, but give it 5 to 10 years, or one of you guys go get it into this, and that could change. Okay, there are some different theories out there. I don't know the right answer. I am going to try to give you these are the T cells you have to know. These are the B cells you have to know. How exactly they become that, how exactly are they always that, there's some gray area here. So if you get a little confused, we can talk about it in lab tomorrow, or let's have an appointment, a few of you guys, and let's talk about it, and let's do it where we can kind of flesh out and kind of hone in on what we really need to know, and then what is kind of this gee whiz Dr. Granier's expanding and giving you some potential theories that might be out there on how the system works. And again, it's not very well written on this chapter, because this is the chapter where I could be teaching you something, and then in 10 years when you're in graduate school and you're getting your nurse anesthetist or your nurse practitioner degree, you're going to find out, oh, Dr. Granier was all wrong, and so was our book. Okay? All right, so T cells. There are T cytotoxic cells. There are T helper cells. There are T suppressor cells. There are T memory cells. Okay, so over here on this slide, and then again in your slides, there are these categories of T cells, and those are the T cell categories you need to know. All right, so T cytotoxic cells, which back in my day, we just called them killer T cells. They were just killing cells. These cells are going to be activated. That's some of the gray area. They get turned on and they get told, look for this pathogen because it's displaying this antigen. So it's kind of like this. Look for Cubs fans. How do you know they're Cubs fans? They're wearing Cubs logos and clothes and gear. If you see a Cubs fan, kill it. All right? So look for odd cells that are Cubs fans wearing Cubs gear. Look for cells that are self, that belong to us, but they're wearing Cubs gear, and kill them all. Kill anything that's wearing Cubs gear. Okay? Something had to tell it the Cubs gear is the trigger that you're looking for, the trigger to confirm that you're killing something foreign or something that has invaded your host and is making the host now produce foreign molecules and materials. Okay? Cytotoxic cells want to kill Cubs fans. There you go. All right? They will only kill the fans if they're actively displaying the logos, the antigens. All right? So one of the ways to get around cytotoxic T cells or killer T cells is I could be a cell that has chickenpox virus, but I'm not actively expressing the chickenpox viral proteins and the chickenpox viral molecules on my membrane to tell the cytotoxic T cell that I'm a Cubs fan. I'm keeping it inside. All right, so cytotoxic T cells are going to be my killing cells. They are going to kill any foreign cell that is wearing the wrong antigen. They will kill any of my self cells that are displaying the wrong antigen. And they are looking for that one antigen that they have been sensitized to look for. All right. So again, if I have a cytotoxic T cell, one of them is only looking for Cubs fans. I could have another pathogen that's a Royals fan, but I have to have a different cytotoxic T cell looking for the Royals gear, looking for the Royals logo, and killing those fans. Okay? So that is how the cytotoxic T cells kind of work. Okay? Now, suppressor T cells are trying to keep the cytotoxic T cells from going a little overboard and start to kill inappropriate cells, like Cubs logos are blue, St. Louis Blues logos are blues. Let me start killing these guys. No. Cytotoxic T cells, if they start to go a little too crazy, suppressor cells are meant to kind of keep them in check. They are trying to help, again, keep the cytotoxic T cells from only killing the cells they have been designed to find and kill. Okay? Now, helper cells. And again, if you really get into the literature, it is way more complicated than what we're going to present here, but we are going to try to keep it simple. Remember we talked about macrophages, neutrophils, eosinophils, and dendritic cells, all of these phagocytic cells that are going to be part of our nonspecific defenses. Okay? They are going to take in foreign material, digest it, and they're going to take some of that foreign material and they're going to show it to T helper cells. The T helper cell is going to be the cell that says, you just ate a bunch of organelles from an exploded cell. No big deal. Or the T helper cell is going to be the immunocompetent cell to say, oh, this is fungus material. This is not self. This is good. This is something that tried to use our cut, our opening to get in and is trying to take up residence inappropriately. Okay? So your T helper cell is the cornerstone cell for our purposes. It is the cell that gets the information from your phagocytic cells that are non-specific defenses and tells the specific cells, naive B cells and naive untold yet cytotoxic killer cells, to come over here. This is your new target that we now know has gotten into our system. Now go find it, tag it, and kill it. All right? So the T helper cell is the cell that kind of bridges what's happening in the nonspecific defenses and bridges over cells that have yet to, again, be sensitized and said, here's the Cubs logo, go kill these cells. It's saying, here's the Cubs logo, or here is, let's try to use a different analogy. Here's the chickenpox virus. Here's what it's going to look like when it shows up on a cell. Go find any cells that are displaying chickenpox virus and kill them. For B cells, here is this chickenpox antigen that's going to show up on cells. Let's make a tag that we can just spread out and it can try to find all these cells that are holding that. All right. So the helper T cells are kind of the link between the monitoring cells up front of phagocytosis and turning on specific defenses. And we know that because of HIV. All right. Helper T cells are the HIV target cell. That's the cell that the virus wants to insert itself. It wants to turn this cell into an HIV making factory. And if the T helper cell is all about making more HIV proteins and particles, and then it explodes and it spreads out those particles to go find more T helper cells, there are no T helper cells for the macrophages and the neutrophils to come over and show, hey, I found pneumonia bacteria. Hey, I found streptococcus. There's no T helper cells to go, oh yeah, this doesn't belong. Let's get some antibodies and let's get some cytotoxic T cells to kill them. All right, so that's where we really learned what T helper cells were. And again, it's a little more complicated than this because there are different forms of T helper cells. But for the most part, I want you to think of the T helper cell as the cell that you bring from your macrophages, from your neutrophils, from your eosinophils, from your dendritic cells. Any phagocytic cell that's eating particles and contents that could have gotten into the body inappropriately through a break in the membrane or a break in the skin, those cells, when they eat things, are going to bring them to the T helper cell. And the T helper cell is going to go, bad pathogen, thank you for finding, or it's going to go, you just ate some debris. No big deal. It's all self. When the T helper cell says this is bad stuff, it then brings over, so it kind of brings together B cells and about to turn on T cytotoxic cells, and it says, you see this little piece of dirt, go find this piece of dirt everywhere and kill it, and B cells find this piece of dirt and put a tag on it with an antibody. Okay? All right, y'all are asking a bunch of questions. Okay? All right, so the T helper cells are, again, kind of the bridge to let us turn on the specific immune system if we find a new pathogen that has shown up and has been eaten by macrophages. Okay? And that's how I want you to kind of think of it. Is it possible the T helper cell could bump into a pathogen like tuberculosis bacteria that hasn't been eaten yet? Yes. And could it turn on and bring over B cells and T cells to say, hey, this guy is here and I know he doesn't belong. Like, this is a crasher to our party. Yes. But the odds of that happening are a little less because the T helper cell is in the middle of the lymph node. The T helper cell is not always maybe at the front lines of where the pathogens might be coming into the system. And so the more likelihood is, hopefully, those pathogens are eaten through phagocytosis and brought to the T helper cell. Not that the T helper cell ends up running into them. Okay? Okay. All right. Something about if it tags and it says, hey, new killer cell, you don't have a job yet. Your job is now to find the chickenpox and kill it. Yes. All right. Yes, that is correct. All right. And it's going to turn on some suppressor cells to say, don't let the killer cells go too crazy. And then there's a memory cell that is going to develop to say, okay, you guys take care of this right now, and I'll keep a watch for this pathogen for the next 5 to 10 years. Okay? Now, that's how I want you to think of these guys and their roles. Okay? And I want you to remember that anytime I turn on a killer cell, that killer cell can divide and multiply and turn into, through mitosis, 2 killer cells. And those 2 killer cells can turn into 4 killer cells. And then those 4 killer cells can turn into 8 killer cells. And so I can very quickly, through a few mitosis cycles, I can try to get a little army of killer cells. And one theory is that as I'm creating this army of killer cells, every now and then one of them becomes a suppressor cell. And every now and then one of them becomes a memory cell. Okay? And so that initial T cell that I brought over to my helper and said, hey, you're looking for Cubs fans, you're looking for chickenpox virus. He could go through 2, 4, 8, 10, 12 mitosis cycles and lead to 10 killer cells, 2 or 3 suppressor cells, and 1 or 2 memory cells. Okay? No, this is for any pathogen. So if my pathogen is a fungus, then my killer cell can be brought over by the T helper cell to say, hey, the macrophage ate this fungus. Now I want you to go find any place this fungus is living in. This fungus is trying to attach to, and I want you to kill it. And again, my cytotoxic T cell could go through some mitosis, lead to 10 to 15 killer T cells, 2 to 3 suppressor cells, and 1 or 2 memory cells, and then goes and tries to find wherever that fungus might be living. Okay? Same thing if it's a bacteria. Let's say it's It's tuberculosis. All right. So my macrophage, this case, maybe it's a pneumocyte dendritic cell in my lungs. It eats the tuberculosis bacteria. It brings it to the T helper cell. The T helper cell says, yep, this is not self. This is a bacteria, a foreign entity. It brings over a T cell that becomes a sensitized T killer cell. It goes through a few rounds of, again, mitosis. It leads to a little army of cytotoxic T cells, maybe 10 to 15 of them, 2 or 3 suppressor cells, 1 or 2 memory cells. And then they deploy out in and around the lungs. They deploy out in and around the connective tissues of the airway. They deploy out to connective tissues in the heart, to the skin, and they are looking for tuberculosis bacteria. Okay? So that is how, again, usually there has to be a T cell, and it's usually that helper cell, and it realizes, hey, this is something that's not self, and it brings over another T cell, and this other T cell then can become the T cell that is cytotoxic, suppressor, and memory. Or in theory, it could be this helper cell brings a cytotoxic, a suppressor, and a memory, and they all then work together to be, okay, now we're dedicated to this pathogen as our mission. And I'm forever going to be the tuberculosis group. And then I have a group over here that's my chickenpox group. And I have a group over here that's my streptococcus A group. Okay? So that's how that works. All right, on the B cell side, B cells stayed in the bone marrow to finish their maturation. Okay? So again, the T helper cell gets introduced to a pathogen. It's going to call over one of my T cells to turn into my cytotoxic suppressor and my memory cells. It's going to call over a B cell that hasn't been sensitized to anything yet. And when it calls it over, like the little picture shows right here, the B cell and the pathogen and the antigens of that pathogen in some cases, because the pathogen might actually be destroyed and digested up by the macrophage, all right? Those antigens are going to be presented to the B cell. Now, here's the weird part that I don't understand and I don't have a good answer to. In the gene, in the DNA of the B cell, there are some areas for the antibody that has introns and extrons. So it has these areas of DNA that are kind of like filler DNA, and then it has areas that could be, again, manipulated into being the antibody that we, then know as immunoglobulin, big letter G, immunoglobulin, big letter M, immunoglobulin, big letter A. Okay, and depends on which extron or which little piece of DNA is put or selected to be part of the antibody. All right, so somehow the B cell, when it gets introduced to the antigen, it has some ability to go into its DNA and cut out and splice some of the useless parts of the DNA that are not going to help it create an antibody that will always specifically target and, like a puzzle piece, find this antigen and bind to it. I don't understand that process. I don't need you to understand that process. But when a B cell is introduced to the antigen of a pathogen that it's going to sensitize, it is going to manipulate its DNA. And if it had the ability to make IgG, IgM, IgA, IgE, at the end of the sensitizing process, it is only going to make one antibody. And it's going to make that one antibody that is the perfect fit for this antigen. Okay? The T cells, I don't know, Portia. I don't know if the T cells have any influence on this. It's possible that the T helper cell, when it pulls the B cell over, maybe through the cytokine communication, maybe it influences the B cell that you're going to make IgE against this pathogen. Or maybe it can tell the B cell this is kind of bacterial. I don't know. That's a great question. That's a PhD work right there. Does the T cell influence the B cell, the T helper cell influence the B cell, and how it's going to manipulate its DNA when it creates an antibody for an antigen? I have no idea. but the B cell, which was considered naive because it hasn't been exposed to a pathogen yet, when it's exposed to a pathogen, it is going to manipulate and fix its DNA to where it will only make one type of antibody, and those antibodies are shown with these little Ys, all right? And it's going to go through a cell division process, and when it goes through mitosis, one cell will become a plasma cell initially, and one cell is going to be a memory cell. And the memory cell, again, the DNA in there is always going to have that genetic code for this antibody that will always find the antigens on tuberculosis or the antigens of the polio virus, all right? And so that's how you end up with that lifetime immunity, because those memory cells exist. All right. And then this plasma cell, also known as a clone cell, it'll go through mitosis again, and then it'll go through it again and again. And I will end up with a little army of maybe 10, 15, 20 B cells that are actively making a bunch of antibodies that are specific to my target, my pathogen. And the thing about antibodies is they are small proteins and they're water soluble. So they more easily move in blood than a cell does. They more easily move in and out of my blood supply because they can fit when blood is forced out through filtration. They can fit through those pores with glucose and with sodium and end up in my interstitial fluid and around cells. They can easily flow into my lymphatic area. They can easily flow through my lymph nodes, right? They're kind of like little glucose molecules. They're really small, and I can create, again, a big horde of these molecules. And if I'm 70% water, this big horde of molecules can more easily disperse and quickly get into all the nooks and crannies where I have water in my body. And when they attach to the pathogen, all they do is attach. And if you attach to the pathogen, they are going to make the pathogen a bigger target. And so if the pathogen was something small, if the pathogen was a really small little bacteria or a really small little viral protein and DNA, RNA material, it's going to make it a bigger molecule because now it has a few maybe antibodies attached to it. And now it's harder for that pathogen to evade and not get captured by macrophages, dendritic cells, neutrophils, eosinophils, and it's easier for it to potentially be eaten up and destroyed. Okay. But the one thing you have to remember is the antibody does not kill anything. All it does is attach to cells, attach to molecules, attach to fragments of things that are displaying the antigen that the T helper cell made the B cell sensitized to, because the T cell determined it was a non-self protein. Okay. So if the T cell is wrong, because I think Corey or somebody asked about immunosuppressants or autoimmune. If the T cell chose an antigen that is actually self, and now you have this antibody army binding to Schwann cells on all of your peripheral neurons, and all of your Schwann cells are now being targeted to be destroyed. Yes, that could be a bad problem. So the whole system is the T helper cell that brought the B cell over and told it to sensitize to this protein, glycoprotein, phospholipid, phosphoprotein, lipoprotein, whatever it is, that the T helper cell is correct in its assessment that this is not self. This is from a foreign particle, a foreign object. And we do get it wrong. Okay? Sometimes it's foreign, like it can be a piece of pollen, and our immune system can overregulate and upregulate and cause, inflammation and swelling and damage to occur inappropriately to something that, yeah, it was a little piece of pollen, but it wasn't going to hurt you because it was like a dead piece of fragment. Okay? It wasn't an active living pathogen trying to propagate. All right? Now, one of the big questions is, could the B cell interact with a bacteria, with a virus, with a fungus? Could it interact with it and turn on without the T helper cell? I don't know. It's possible, but the original theory is that the T helper cells and the T cells going to the thymus gland, they are the immunocompetent cells. So they're the cells that know if something is self or not self. The B cells stayed in the bone marrow, so they did not gain this immunocompetence because they didn't go live in the thymus gland and get exposed to thymolytic hormones. Okay? And when I learned this, the B cells did not turn on unless the T helper cell introduced them to a pathogen and confirms that this was a pathogen, and yes, it's okay to make an antibody against it. All right? So that's the theory that I grew up under. It could be wrong. It could be that maybe the B cells could potentially turn on and become sensitized to a pathogen without the T cells involvement. But I don't know that for sure. So just to be safe, think of B cells as cells that can make antibodies, and antibodies give us more ability to spread out and find our pathogens easier with a small molecule than waiting for cells to find it. And they can help find pathogens that are hiding and make them bigger targets. And usually that process, for most of our purposes, is going to always be initiated because a T helper cell introduced the pathogen to a naive B cell. Okay? Now, because again, there's a lot of mitosis occurring, when we have naive B cells turning into, again, rapidly mitosing and plasma cells, that's usually going to be part of the reason why we think our lymph nodes, when we're sick, they're getting bigger. There's a lot of cell cycle and mitosis occurring. Well, some of the cells that are going through that cell cycle and mitosis are all these B cells that are now ramping up to form a little army of antibody making cells. The other reason why you're going to have a bunch of swelling is as the number of antibodies in that lymph node increase, antibodies are like a solute. And so more water is going to be drawn in there, and that's going to make, again, a little bit more swelling. Okay, now the antibodies are going to leave with the lymph fluid and expand out, but the number of those plasma cells, those germinating, going through mitosis cells, is going to increase. Some of it's because of the B cells, and again, some of it could be because our T cells, remember the cytotoxic T cell, I make one cytotoxic T cell and it'll go through mitosis and it will make 2, and then those 2 will go through and make 4, and then those 4 will go through and make 8, and then 16. All right. And so again, some of the reason why our spleen and our lymph nodes, and when we are actively fighting an infection, the number of the B cells in your node, the number of your T cells in the node, and then the spreading out of those, and the number carrying white blood cells, lymphocytes in your blood, all go up. And you can sometimes see that with, again, your lymph nodes are swollen, and when you palpitate, they become a little bit more enlarged. You open your throat and you go, ah, and your tonsils are more inflamed, more big, more robust. Okay. Some of that is because there's more particles. Some of that is because there's more antibodies. Some of that is because there's more cells going through cell cycle, mitosis, dividing, and differentiating. Okay. All right. Natural killer cells. Okay. Natural killer cells are newer cells because when I went through this in the 90s, they were not in my textbook. I never heard of a natural killer cell until I started teaching in around the year 2004. Okay. So, natural killer cells. They are immunocompetent cells. They did not go to the thymus gland. So, they are cells that stayed in the bone marrow to develop. They did not go to the thymus gland. They are immunocompetent. So, this confirms a theory that you can become an immunocompetent cell and not be a T cell and not go to the thymus gland to develop, because the natural killer cells are these cells. Okay, now natural killer cells are going to be part of non-specific immunity. So T cells and B cells tend to be part of specific immunity, and the T helper cell is the bridge. All right, natural killer cells are part of non-specific immunity. They are going to circulate in your connective tissues, in your body water. So they're not going to try to house themselves only in the lymph nodes, only in the spleen. They are going to go out and try to interact with all the cells of your body. Okay? So they're like door-to-door salesmen. They are going to try to go to every neighborhood house in the neighborhood, and they're going to try to knock on every door. And when they knock on every door, they're trying to confirm you belong. You're a house that belongs. All right? So you are people living in this house that have the right genetic makeup, that you are self. And then they're going to look at, are you doing things appropriately? Meaning, are you going through normal cell cycle? Are you, if you're an epithelial cell, are you doing epithelial type cell functions? If you're a gland cell, are you making the right amount of hormone or the right secretions and doing what you're supposed to do? All right, so one of the things the natural killer cell does is first say self or not self. If you're not self, it kills you. If you are self, the natural killer cell is then going to go a step further and try to determine if you're doing things appropriately. And by doing this check, it can see if you are hijacked by a virus, hijacked by some other pathogen that's using the cell as a host to try to then use the ATP and the glucose and the fuel to run their own life cycle and not necessarily the cell's life cycle. All right. So if you are infested by some type of pathogen and you're not doing what you're supposed to do, the natural killer cell can then decide to kill you, even though you're self. The third thing the natural killer cell is looking for is yourself, but now you've gone crazy and you're beginning to do inappropriate things. So one of the examples I say is in your genetic code, you have a gene for fetal hemoglobin. Well, the only time you should ever make fetal hemoglobin is if you are a red blood cell for a fetus and you make fetal hemoglobin to be able to get oxygen from mom and deliver it to cells. So if you are a pancreatic gland cell, epithelial cell, and the natural killer cell walks up to you and says, "Oh, yourself, what you doing?" And you're like, "Oh, I'm making some fetal hemoglobin, and I'm about to go through mitosis." The natural killer cell can be like, "Whoa, buddy, you're going crazy. This is not something you should be doing." And then the natural killer cell could potentially destroy you because you've turned rogue, you've turned inappropriate, you've turned into cancer. Okay? So that's kind of what the natural killer cells function in. And again, when I learned this, I thought that the way I was taught is cytotoxic T cells did this. But what we've learned is, nope, there is this separate cell that is going before I even know there's a pathogen and doing surveillance on my neighborhoods, on my cells, and checking them for self, not self, okay, immunocompetence, but also checking them for, are you invaded? Are you being hijacked and taken over? Okay. And then, are you doing what you're supposed to do? If you are not doing the normal cell cycle, you have turned into a cancerous cell, then I can also kill you. All right? Okay. And the way they kill is a little different than usually how cytotoxic T cells will kill. Okay? So that's why they're now a separate group, and then we dig in, and they don't ever go to the thymus gland. All right, so remember those memory cells. Okay, because of memory cells and because B cells and T cells both create memory cells against a pathogen, we know that some cells could live potentially up to 5 to 10 to 15, even as long as 20 years. All right, now usually what's going to happen is let's say I get streptococcus. All right, I get strep throat. And my body went through and it made some memory B cells and it made some memory T cells. And a year later, I get it again. When I get it the second time, my memory B cell has the antibodies on there. And those antibodies all of a sudden bind to my pathogen. And boom, this cell goes, hey, our pathogen's back. And it starts going through mitosis. And it starts going through and creating more plasma cells. And then at some point in time, it creates a new memory cell. Okay, so when my original memory cell only lived a year, 2 years, I had another interaction with that pathogen. The memory cell ramped up my system again, and eventually I created a new memory cell that is now going to be the surveillance for the next 2 to 4 to 5, 10 years. Okay, and so this is part of having those memory cells, how we all got inoculated for, let's say, polio. And so we all should have these memory cells for polio in our system. And it might be that during at some point in our lifetime, maybe some of us did get exposed to polio virus, and those memory cells actually propagated and developed the antibodies and cytotoxic T cells and helped us kill and get rid of polio before we really realized that we were sick with polio. And we probably just had like a little minor 1 to 2 day, cold and fever. And, we just said a 24-hour bug. And we created new memory cells. And so it's possible that this happens to us all the time. And we actually are fighting off some of things that we've been inoculated to, and we're creating new memory cells. And so even though our memory cells might only live at best 20 years, we live for 80 years, and we get lifetime immunity against certain diseases and conditions. And it's because if and when we do ever find ourselves with those diseases and conditions attacking us, those memory cells ramp up, create a defense, create new memory cells, and then we let them go for the next 5 to 10 years. Every 5 to 10 years, I think we normally have to get a booster of tetanus. So in the event that we worry that the memory cells might not be there, well, we booster ourselves and restart the system on purpose to create new memory cells, create new antibodies, create new maybe T helper cells, bringing new T cells and B cells over to the tetanus material and creating new memory cells. All right. So smallpox. When I was in the Air Force, so back in 2002, 2003, I was living in Japan, and they were making people who potentially go to Korea, if, the North Koreans jump the wall tomorrow, that they worried that North Korea and China and Russia would release smallpox to try to decimate the population. And so everybody who didn't have a smallpox vaccine was given the smallpox vaccine before especially if you were in this combat job. And those people, because there were still some people who had gotten a smallpox vaccine before, so they had the little indentation. Some of you are too young to remember your grandparents, all right? But they already had their little indentations saying, I have had smallpox vaccine before. They were given a booster. All right. And so again, when you get the smallpox vaccine, they put this little fork with prongs and they poke you about 4 or 5 times to make sure the virus on the prongs gets into the dermis, hypodermis layer and gets involved and maybe, attacked by some macrophages, neutrophils, and then is presented to T helper cells and then initiates T memory cells and and B memory cells against the virus. If you get a booster, they have to puncture your arm 30 to 40 times. Yeah, so it's like 10 times more punctures because they want to make sure that if you have memory B cells around, they ramp up and you create new potential cells as well to the viral load. All right? So again, if tomorrow they decided, hey, all of us who had the polio vaccine back in the 70s, 80s, and 90s, we all need a booster, they could give us a new booster. Again, that's going to try to create more memory cells and B cells and T cells. And if there are memory cells already in us, maybe ramp them up a little bit to help create some new fresh ones to live the next 5 to 10 years. Okay? So that's kind of how that works. All right. Going back to how do we create lymphocytes? Remember, everything starts in the bone marrow. And remember, it was like IL and EPO and colony stimulating factor. That's how you got the myeloid progenitor cell. And the myeloid progenitor cell gave us eventually either red blood cells, platelets, neutrophils, eosinophils, and basophils, okay, and macrophages. All right. Remember the other side of the equation, you don't have TPO, you do not have EPO, you do not have colony stimulating factors, or they're not in high amounts. We have different cytokines, different interleukins, different signaling molecules that turn our hematopoietic stem cell into a lymphoid progenitor cell. And quite a few of you missed this on the test. I think that was one of the questions of like, which of the following would not give rise to some of these cells? And I think it was like, you shouldn't have a lymphoid progenitor cell give you a macrophage, or it was some kind of question about this. All right, so this concept is here. So now the lymphoid progenitor cells, again, we now know, and this is a little different from what chapter 19 showed us, we now know that the lymphoid progenitor cell can give us what can become T cells, and those T cells at a certain point in their production leave the bone marrow of your epiphysis, leave the bone marrow of your sternum, leave the bone marrow of your hip bones, of your vertebra, wherever your skull, wherever that wherever that spongy trabecular bone is located. It leaves those areas and goes to the thymus gland. In the thymus gland, it gets sequestered in this little pod area with these epithelial cells around it. And those epithelial cells secrete molecules and hormones into this pod. And that area is where the T cells finish becoming immunocompetent and mature. And then when they're ready, they move back into the blood supply, and they can now hang out in the blood supply, take a ride in the blood supply to lymph nodes, to the spleen, to the liver, to the connective tissues, the skin, the hypodermis, wherever they're going. And they can go and take up residence in their spots. Okay? The cells that stay, the lymphoid progenitor cells, can become either B cells or natural killer cells. All right? And when they get to a point where they're ready to leave, remember the natural killer cell can leave, go in the blood supply, and go anywhere, and it's going to start kind of like, okay, I'm going to take the kidney, and I'm going to go through, and I'm going to touch every cell in the proximal convoluted tubules of this region of the kidney, and I'm going to make sure every proximal convoluted epithelial cuboidal cell is doing what they're supposed to do, is self, and is not hijacked. And anyone that is not doing what it's supposed to, is hijacked and not self, I'm going to kill. Okay? The B cells, again, this is a little bit of that gray area. The B cells are going to leave the bone marrow, and they are going to go to potentially the tonsils, the lymph nodes. They can stay in the blood circulating, the spleen, the malts, the pulse, the wherever. All right? But they are still a step away from maturity. They have not fully turned on, because remember, B cells do not turn on until you introduce them to the pathogen and the antigens that you want them to start making antibody against. So even when the B cells leave the bone marrow, they are ready to go. They just haven't been introduced by a T helper cell to be, here is your antigen, here is the pathogen, the antigens of this pathogen. These are the molecules we want you to make antibodies against, and then turn into mitosis and start making a little army of plasma or clone B cells to make a bunch of antibodies, and then a few of your offspring can also become memory cells. Okay, so the B cell, and again, when we look at the lymph nodes, they sometimes, when they're still naive, they are going to hang out in the medulla or the center of the lymph nodes. And they're going to hang out there because if a pathogen comes in to the lymph node and we've already established that we know this pathogen, antibodies and cytotoxic T cells should quickly be able to attach and start killing it. If it's a new pathogen, we're hoping that when it comes into a lymph node, macrophages, dendritic cells eat it. And they might not, but we hope they do, and bring it towards the middle of the lymph node to show the T cells, the helper T cells, hey, we ate this. It came into the lymph node. And then the lymph node or the T helper cell can real quickly say, hey, B cells, hey, a few of you naive T cells, come on over. Here's your new pathogen and antigens we're going to start targeting and ramp up a defense against. And the ones that are already, again, sensitized, those B cells making a lot of antibodies, those memory cells, they live in the cortex. All right, so when you're reading about the lymph nodes, that's kind of how some of the setup of how things happen is because of which cells live where and why they're there. Okay, now I've given you a lot of information on cells today. This is material that you can review. We'll go through it again. This is lab. This again is a little bit of what I'm talking about of how the lymph node is organized. Remember that lymph nodes are organs. So the outer third is like a kidney, like the adrenal gland. We're going to call that the cortex. And then the middle, we're going to call the medulla, right? And again, we're going to have little push-ins of reticular tissue to make lobes, so like pizza slices. And when our afferent vessels bring fluid in, that fluid's going to spread out. And again, that fluid spreads out, and we hope it gets eaten by some of these, again, dendritic macrophage-like cells, right? And then as it's trying to move towards the exit, and we usually have one exit, okay? So if a cell comes in like right here, and here's the exit, it's got to kind of like maneuver its way through these dendritic, these macrophages. It's got to then get through germinal cells, cells going through mitosis, cells going through lots of cell cycle and cell processes. So this is where you have a lot of plasma B cells, you have a lot of antibody making cells and a lot of antibody. And you got to make it through this space that's full of antibody, full of B cells going through a turnover. And if these guys don't eat you, if antibodies don't stick to you, and you make it towards the center and the T helper cells don't randomly bump into you, you might make it through this lymph node. But if you're something that, again, the dendritic cells eat, the macrophages eat, and the antibodies bind to, the goal would be by the time you got to the center region, the T helper cells would be like, oh yeah, dendritic cell, you ate something, yes. Bring a B cell over, bring a naive T cell, let's start up a new defense system. Or, hey, who are you? You're not supposed to be here. And then let's bring some B cells over, let's bring some T cells over, and let's start up a new defense. Okay? So, end of the system, all of these cells have their roles. All of these cells are doing their little piece and part of the puzzle. And because of that, the fluid that we take out of interstitial system and put in the lymphatic vessels, and we're working to get back to the thoracic duct and the right lymphatic duct. That fluid has been through potentially so many lymph nodes, so many traffic circles, so many stoplights, that if there are antigens and if there are cancer cells and if there are invasive pathogens, we have hopefully found them, tagged them, and removed them. And so if something is to be a blood type infection, the goal is that if it started from a cut in the skin, it's not going to turn into a blood infection overnight, because we have this system that keeps pathogens from very easily getting into our blood supply, okay, and getting the ability to spread out and take over massive major organ systems far away from its entry point. All right. And usually when something tries to get in, whatever area that's closest to the entry point, so if it's a finger, chances are the lymph nodes in the armpit and the maybe the armpit and the triceps area are going to be the ones to probably be the early warning for that pathogen. If it tries to get in through the nose, mouth, ears, the lymph nodes and the tonsils in the neck and the throat region are going to be our early warning system. If it's trying to get in through the stomach, the small and large intestines, because it got, with food digestion, it was embedded in the food, then potentially the lymph nodes in the belly, the malts, the palps, Peyer's patches, they are going to be the ones to find it. Okay. And so that's same thing with cancer. If a cancer is going to originate from potentially skin in my fingers or bone in my fingers, then my early warning lymph nodes are going to be maybe the ones in my armpits. If a cancer is going to try to develop from my ovaries, from my bladder, it might be that the groin, the vertebra, and lymph nodes in and around the abdominal pelvic region are going to be the areas that are going to catch that cancer before it gets dispersed and tries to move elsewhere. Okay? And so that's part of the reason why something like breast tissue, if a cancer starts in the breast, we tend to tank the lymph nodes in the armpit, in the chest area, even up into the neck region, because those would be the potential lymph nodes that cell of cancer leaving and migrating away would potentially get first caught up in, slowed down, and stopped. All right, and would start to tell us if the cancer cells are beginning to try to migrate. All right, I'm going to stop the recording here because that's a lot of information. And some of this, again, is information for your lab that we'll go through tomorrow.